Hi, and welcome to the tutorial for problem 1 of practice problem 2-4 for BIOS E1A. This question states, you have 3 partial diploid strains of E. coli that contain different mutations within the lac operon. Wild-type genes are denoted with a superscript plus on the gene symbol. Mutations in genes that lead to defective or absent gene products are denoted with a superscript minus. Assume genes not listed are wild-type. For each genotype, determine whether the expression of beta-galactosidase will be inducible, constitutive, or entirely lacking. Briefly justify each answer. It then lists 3 genotypes of partial diploids to analyze in parts A through C. Part D asks about beta-galactosidase activity for wild-type bacteria grown on different media. And Part E asks about what would happen if the bacteria were grown on the same media as in Part D, but could not synthesize cyclic AMP. In order to answer these questions, all we'll need to do is review the mechanics driving the lac operon to either be active and transcribed or to be inactivated. Here we see the lac operon boxed in red. The operon is responsible for allowing the cell to digest lactose. Now, E. coli actually prefers to use glucose as an energy source, but when glucose isn't present or it's scarce, this operon is activated and the cell can digest lactose for extra energy. Upstream of the lac operon, we have another sequence that codes for a repressor protein, boxed in blue here. In a wild-type cell, this gene is constitutive, meaning that it's always being expressed at relatively constant level. This means that there is always going to be a repressor protein present in the cell, given a functional version of this gene, which we label here as I, ensuring that the cell doesn't wastefully transcribe the genes of the lac operon when there is no lactose present. Now we'll move over to the lac operon. The operon consists of a promoter, operator, and 3 structural genes labeled and highlighted here as Z, Y, and A, short for LAC-Z, LAC-Y, and LAC-A. These 3 genes each code for proteins that allow the cell to digest lactose. They each code for a different protein that helps in the pathway. You can see each of their protein products here. Remember that LAC-Z codes for beta-galactosidase because that will become important later on. That's what the question really asks about. Moving to the start of the lac operon, we come to the promoter. Because RNA polymerase can't bind directly to LAC-Z, LAC-Y, and LAC-A and start transcription, the promoter is necessary as the site of RNA polymerase binding. Without a functional promoter, RNA polymerase can't bind and none of the structural genes are going to be transcribed. Next, we come to the operator. The operator is the site that the repressor protein will bind to. When repressor protein is bound to the operator, RNA polymerase can't bind to the promoter, and transcription of the genes of the operon won't work. So, given that repressor protein is always going to be present in cells with a working repressor gene, the default of this operon is actually in an inactivated state. This makes sense because if the cell has no use for the gene products that the operon produces, that is, there's no lactose around, it's wasteful to be transcribing these genes. You'll just get transcripts that code for proteins that aren't going to help the cell. So how does the lac operon become activated? Well, the operon is only useful if there's lactose present for it to break down. If lactose isn't present, the repressor is bound to the operator and transcription doesn't occur, as shown here. When lactose is present, the cell needs to be able to break it down with the gene products of the lac operon. In order for this to happen, lactose, after having been converted into an isomer called allolactose after entering the cell, actually inactivates the repressor protein, and it no longer interferes with transcription. This means that LAC-Y, LAC-Z, and LAC-A genes are free to be transcribed and to break down lactose. But even when lactose is present, the cell prefers to use glucose as an energy source. It would be wasteful to be breaking down lactose at such a high rate when there is glucose available that the cell prefers. So how does the cell ensure that it's preferentially using glucose when both glucose and lactose are present? The answer is that in order for lactose to actually be broken down efficiently, another protein, cyclic AMP, receptor protein, which we'll call CRP for short, must also bind to the lac promoter. As we can see in the situation here, CRP is inactive when there's a high concentration of glucose, even when lactose is present. So you need the CRP also to activate the lac operon. When glucose levels fall, as demonstrated here, the cell needs to utilize the lactose around it for energy. A shortage of glucose increases the amount of CAMP in the cell, and then this CAMP is able to bind to CRP, and that activates it. Upon activation, this CRP complex, CRP combined with CAMP, binds to the promoter of the lac operon, and this ramps up transcription of the lac genes to high levels. So it's under these conditions, shown here, that we get high levels of transcription. CRP is bound to the promoter and the lac repressor has been inactivated by allolactose, so it's not bound to the operator. With this information, we're ready to tackle the question. So let's look at part A through part C first. These questions ask what the expression pattern of beta-galactosidase encoded by the LAC-Z gene would be in partial diploids with certain mutations. In part A, we see that the individual has one functional copy of the repressor gene, which we call I in this case, that's linked with a non-functional operator, which we call O, and functional LAC-Z gene, Z. The other operon present has a non-functional repressor gene, a functional operator, and a non-functional LAC-Z gene. So that will look something like this. If we look at the top operon, we can see that it has an active repressor gene, but there's no operator for that repressor to bind to. Thus, even in the presence of repressor, LAC-Z transcription will be uninterrupted, meaning that production of beta-galactosidase will be constitutive. It's always going to be happening. The bottom DNA strand has a non-functional copy of LAC-Z, so it won't produce any beta-galactosidase, because that's the gene that actually codes for the beta-galactosidase. If that's not functional, then you're not going to produce any. Now we'll look at part B. In this situation, we can see that both repressor genes are non-functional. This means that no repressor protein is being produced, and even the DNA with a functional operator can't be repressed. The top strand is fully functional other than this one mutation, so expression of beta-galactosidase is again constitutive. Moving on to part C, we can see that the bottom DNA strand has a functional repressor gene, so repressor protein is going to be present in the cell. Because the top strand has a functional operator, production by that strand is inducible. When there's a repressor around, it's going to be turned off, unless something is there to induce it, namely allolactose. The bottom strand doesn't have a functional operator, so production of LAC-Y and LAC-A in this case would be constitutive because there's nothing for the repressor to bind to. But because the LAC-Z gene, which actually codes for beta-galactosidase, is not functional on that copy, it won't produce any of the protein we're looking for. So if the top strand is inducible and the bottom doesn't produce any product, then the whole system is inducible. The next part of this question asks if we expect there to be high beta-galactosidase activity if wild-type E. coli are grown on media containing all possible combinations of glucose and lactose. We know that when lactose is present, or rather lactose is absent, the repressor proteins will be active and bound to the operator, so no protein product will be produced by the operon. Thus, we can look back at our table and say right away that any media containing lactose will not have high beta-galactosidase activity. Recall, though, that even when lactose is present, we also need high levels of cyclic AMP to be present to have transcription to any appreciable degree. CAMP only increases when glucose levels are low. This CAMP binds to CRP to activate it, and the complex then attaches to the lac operon promoter, and this drastically increases transcription. In this sense, it's an activator for this operon. Since beta-galactosidase is a gene product of the lac operon, it will only be significantly produced under low glucose conditions. So going back to our table, we have shown that the only conditions under which beta-galactosidase will show significant activity is one in which lactose is present and glucose is absent. Part E asks us how the answer to Part D would change if CAMP were not present in the cell. As we've discussed, CAMP is necessary for activating CRP to bind to the lac operon promoter. And then this CRP CAMP complex then then binds to the promoter and attracts the RNA polymerase to a more substantial degree than without it. Without CAMP, CRP can't bind to the promoter, and the beta galactosidase gene, LACZ, isn't transcribed effectively. This means that in all cases, the answers to part D would be no without the presence of CAMP. You're not going to get significant transcription of the genes if there's no CAMP available. And that concludes this tutorial. So thanks for watching, and I hope it helped with your studying.